Hello, and thank you for the introduction
and the opportunity to speak today. I would like to start my presentation off by just giving you a bit of background on our research group and the types of research we're interested in. So our MARTA research group is located here in Brisbane in the sunny state of Queensland in Australia. And we reside here at the Translational Research Institute, otherwise known as TRI. Now, TRI is a collaborative institute bringing together 4 partner institutes: the University of Queensland, Queensland University of Technology, MARTA Research, and Queensland Health. And really the idea behind this building was to have a collaborative research environment where improved translation of research and innovative ideas were able to be translated out into improved outcomes in the clinic. And now we're very privileged here at TRI to have access to a range of world-class facilities, including DSPF biological research facilities, gnotobiotics, all the way through to flow cytometry and proteomics capabilities, and right down to through to our clinical research facilities that we have available. Now, as part of the MARTA group, our vision is to enhance health within the community through world-leading research. And how we achieve this is by integrating MARTA research in with MARTA education and MARTA health, allowing for knowledge and practices that come from research outcomes to be translated and integrated into improved healthcare and educational practices across MARTA. And as part of that integrated structure, we have the Queensland Cord Blood Bank that is located here at MARTA also, which is run by Philip Johnson. And so the Queensland Cord Blood Bank was established back in 1998 through philanthropic funds and has since been funded by the government. It's TGA licensed, FACS accredited, and a member of Auscord, and since its establishment has collected over 17,000 cord blood units. This has allowed for over 11,000 units to become made available on the worldwide searchable register for allogeneic hematopoietic stem cell transplant. And more importantly for us for research, this has given the ability for over 3,500 units to become available to MARTA research. And it is really through this collaborative environment and integration of different aspects that's really made the development of our humanized mouse program of research here in the Research Radford lab possible. So through our access to our world-class facilities here at TRI, through to our links with MARTA and our access to the Queensland Cord Blood Bank, it has been integral for the establishment of this program. So our humanized mouse model, we take stem cells that we derive from the cord blood units that we get from the Queensland Cord Blood Bank, and we inject these into neonatal irradiated neonatal immunodeficient mice. And after a period of about 8 to 10 weeks, what we see in our humanized mice is the development of a full complement of human immune subsets within these mice. And we're able to detect human CD45 positive cells across a range of different organs organs within these mice, from bone marrow, spleen, and liver, all the way through in lung and blood. So we've developed our two different humanized mouse models in 2 different strains of mice. The first one is in the NSG-A2 immunocompromised strain, which is transgenic for human HLA-A2, which allows for the antigen-specific anti-targeting within these mice. And also we have the NSG-SGM3 line, which is transgenic for the human cytokines, stem cell factor, granulocyte macrophage, cell stimulating factor, and IL-3, which allows for improved myeloid development within this mouse strain.
Now, the applications that
we have been using our humanized mouse for so far, we've been using them in our research to develop our dendritic cell targeted cancer immunotherapies, where we're taking an antibody that is specific for CLEC9A, which is a dendritic cell surface receptor that is conjugated to a tumor antigen, and using these antibodies to vaccinate people and target dendritic cells to stimulate a person's own immune system to fight the cancer. And we've also been using these models for tumor modeling. So this allows us to look at tumor development in the context of a human immune system and able to treat these mice with therapies that modulate the immune system and see what effect this is having on tumor development. So as I mentioned before, our research group interest is focused on dendritic cells, or otherwise referred to as DCs. These are a rare immune cell type whose role in the immune system is antigen presentation, and these cells can be thought of as the commanding officers of the body's defense system, directing the T cell army where to go and what to attack. And it's really been the rarity of these cells that has really drove our appeal for using the humanized mouse models, because they have given us a viable model in which we can not only test and understand and research the function of these cells, but has also given us a way of developing and testing our new targeted DC targeting therapies for cancer vaccines. So when we do in previous studies, we've done a comparison of these dendritic cells that develop in our humanized mice versus those that we find in human blood, and looking at the expression profile of two different dendritic cell populations from our humanized mice compared to their equivalent human populations. The same human populations derived from human blood, we can see that the expression profiles are pretty close between the dendritic cells that are derived from humanized mice and their human blood equivalents. And this indicates to us that the humanized mice will provide an excellent model for studying the functionality of these cells in a more viable context. Now, also as a part of this research, we have developed techniques where we're able to introduce antigen-specific TCRs into our mice. So we're able to take a lentivirus encoding a specific, a HLA-restricted TCR, and we're able to transduce our human CD34 positive stem cells prior to injection into our irradiated neonates. And what this allows to happen in the developing humanized mouse is we're able to get at the end of this a complement of naive antigen-specific CD8 T cells within these mice, specific for the antigen we're after. Now, an example of this is using our antigen target of NY-ESO-1. We have a lentivirus encoding the NY-ESO-1 specific TCR, which we can use to transduce our cord blood stem cells, inject these stem cells into our irradiated neonates. And what we see in the developing in the humanized mouse, if we look at the T cell compartment here, is a portion of our CD8 positive T cells are dextrom positive, which represents our NY-ESO specific CD8 T cells. And if we take these T cells out and look at them in vitro functional assays, we're able to show that we're able to with re-stimulation with antigen-specific peptides, we're able to generate an interferon gamma response with these cells. And we're also able to show that they have the ability to cause antigen-specific lysis when looking at either peptide-loaded T2 cells for NY-ESO or an NY-ESO positive tumor cell line. So this together gives us an ideal model for testing our antigen-specific DC targeting antibodies and therapies. So in this case, we were able to take, in this example here, we could be able to show that we can take our CLEC9A NY-ESO DC targeting antibody and prime naive NY-ESO1 specific CD8 T cell responses in vivo. So we have our humanized mouse harboring our naive NY-ESO specific CD8 T cells. We can vaccinate that mouse with our DC targeting antibodies along with antigens, adjuvants, and then harvest the splenocytes from that mouse. And through ex vivo restimulation with the NY-ESO peptide, we can show that we can stimulate a significantly increased interferon gamma response in those mice that have received in the splenocytes of those mice who have received in vivo priming using our antibodies compared to controls. So it's really through this research on dendritic cells and development of our humanized mouse program that paved the way for the collaboration between Associate Professor Mark Chong and Professor Kristen Radford here at MARTA Research that drove the research behind the paper that I'm presenting to you today. So Associate Professor Mark Chong at the St. Vincent's Research and the collaboration with Kristen Radford resulted in this article where we were looking at a collaborative project to look at the effects of DROSHA and DICER on human hematopoietic stem cell function. And this paper was really was based off a paper that was by Mark Chong's lab, published in Nature Immunology, where they were looking at the effects of DROSHA and DICER in a pure mouse model of hematopoietic development. And this work was primarily done by Timothy Johansson. So just to give you a bit of background on what DROSHA and DICER do normally, these two genes encode enzymes that have central roles in the biogenesis of microRNA. So they cleave the pre-microRNA stem loop structures to form the final duplex microRNA prior to the formation of the mature microRNA. So they really are essential to the processing and generation of microRNAs in normal in the microRNA pathway. And the aim behind the following project in a pure mouse model setting was to investigate whether this microRNA pathway is required for dendritic cell development. And this was done by this was going to be studied by looking at the ablation of DROSHA and DICER. And so the mouse model that they used in this study was a mixed bone marrow chimera pure mouse model, where an irradiated mouse with bone marrow from mice with tamoxifen-inducible, that's been reconstituted with bone marrow from mice with tamoxifen-inducible expression of CRE, and with bone marrow from a mouse with LOXP flanked alleles, encoding either DROSHA or DICER, that was able to be deleted by the tamoxifen-inducible CRE. And so these mice were were developed and then fed on a treatment of tamoxifen for 3 weeks and then analyzed for their immune development. And what they found from this study was that both DROSHA and DICER had the ability to decrease the frequency of both conventional DCs and plasmacytoid DCs within these mice. However, it was shown that the effects of DROSHA deficiency on DC development was much severer than what was seen with DICER. They also showed that deficiency in development extended to further myeloid populations. However, it did not impact the lymphocyte development within these mice. In vitro studies, they took cells from these chimeric mice, and in vitro, it differentiated those into DCs under dendritic cells under tamoxifen treatment. And these studies showed that while DICER showed no effect in vitro on the development of dendritic cell subsets, once again, impairment of DC development was seen with the deficiency in DROSHA. And by sorting the progenitor populations from the different progenitor populations from these mice, they're able to show that this impairment of DC development from progenitor cell populations occurs very, very early on in haematopoietic development in the LSK cells. And once these progenitors move further along the line of commitment into different subtypes, the effects of DROSHA deficiency on dendritic cell development and frequency was lost. So to further investigate how the what mechanisms are lying behind the effects of this severe response seen with DROSHA deficiency on haematopoietic stem cell development, comparative gene expression analysis was done on haematopoietic stem cells from the DROSHA deficient mice versus the DICER deficient mice. And what this showed was that a number of gene targets were upregulated with the DROSHA deficiency. Now, it was reasoned by Mark John's group that that given the discrepancy between the severity and response between DICER and DROSHA, that this more severe response effect on haematopoietic development in the DROSHA mice was likely to be mediated by a non-microRNA related function. Now, it has been shown in the literature that DROSHA has the ability to cleave mRNAs. So mRNA targets which were shown to be upregulated in DROSHA were then overexpressed in wild-type mouse hematopoietic stem cells, and they were able to show that the overexpression of two particular targets, Mil9 and TOTO1, were able to partially recapitulate the effects that DROSHA was shown to have on DC development in vitro. So as you can see here, once placed into in vitro differentiation, Mil9 and TOTO1 produced a lower frequency of dendritic cell development in these mice, in this culture. So in summary of this background, in a pure mouse model setting, they were able to show that DROSHA knockdown in mouse hematopoietic stem cells severely impacted DC and myeloid development, whereas DICER knockdown only partially impaired this development. This suggested that other mechanisms, in addition to just the effects on the microRNA pathway, were mediating this more severe DROSHA response. In vitro studies showed that this impairment by DROSHA occurred very early on in the stages of haemopoietic stem cell development into other cell types, and that investigation into what mechanisms may be behind this development, they showed that DROSHA's alternative function in the degradation of target mRNAs, specifically the MIL9 and mRNAs MIL9 and TODR1, were absolutely critical for DC differentiation in a pure mouse model setting. Now, given these results that they found in the mouse models, they then wanted to take this forward and look at what effects, look at whether this impaired dendritic cell and myeloid cell development that they saw in the mouse models was able to be replicated in a human setting. And this is where this paper has this is where this paper has developed from that I'm presenting to you today. So in order to look at the effects of DROSHA and DICER knockdown in human haemopoietic stem cells, the first thing that needed to happen is we needed to develop lentiviral shRNA constructs that were designed to knock down DROSHA and DICER. So in Mark Chong's group, they designed 2 shRNA knockdown constructs for both DROSHA and DICER. These were transduced into our haemopoietic stem cells using lentivirus. And we were able to show that shRNA1 for DROSHA and shRNA2 for DICER achieved a knockdown of greater than 70% of their respective targets, either DROSHA and DICER, in the haemopoietic stem cells. We also were able to show that this level of knockdown was sufficient to knock down the biogenesis of microRNAs by looking at a representative microRNA, expression of a representative microRNA in these transduced cells using these shRNA knockdown constructs. So once we knew that that was working, we had sufficient knockdown, and that was able to knock down, and that level of knockdown was actually sufficient to impair microRNA processing. We then took these constructs and generated some virus, and were able to place this in our humanized mouse model. So we used the lentivirus for the shRNA constructs, transduced our human haemopoietic stem cells. We then cultured these for a couple of days in vitro prior doing a GFP sort for positively transduced cells, and engrafting these into our immunodeficient mouse pups to generate our humanized mice. And we are able to look at the haemopoietic effects that 9 weeks later that these shRNA constructs had for DROSHA and DICER had on the development of haemopoietic stem cell, haemopoietic subsets within these mice. So what we saw when we looked at our mice at 9 weeks when they were harvested is we saw severe impairment of haemopoietic stem cell differentiation and development in our DROSHA knockdown mice. So when we looked at the human CD45 reconstitution in these mice, you can see here with the DROSHA shRNA knockdown in both spleen and bone marrow, we have less than 1% of the human haemopoietic stem cells that developed in these mice are positive for GFP, showing that they are positively transduced with our they are positively transduced, and that we get this outgrowth of non-transduced cells. However, when we look at DICER, this had no effect on the percentage of GFP positive cells compared to our GFP control, suggesting that it is not causing any effect on the level of engraftment and development of hematopoietic stem cells, human hematopoietic stem cells within these mice. So to confirm whether this impairment we've seen in DROSHA is happening, is a loss of, is due to a proliferative growth of the hematopoietic stem cells, or whether it's an impairment in differentiation. We also had a look at these cells in vitro. So once again, we took our lentivirus, transduced our hematopoietic stem cells, and sorted our different constructs for their GFP positive cells, and placed these into expansion and then differentiation into dendritic cells cultures in vitro. And what we saw as a result of this is that there was no effect by any differing effects by any of the constructs or either of the two different shRNAs compared to the GFP control at expansion of the CD34s, showing that these cells are able to grow normally. However, once we put them into differentiation, we showed that once again, DROSHA knockdown caused severe impairment of the hematopoietic development and differentiation in these cultures. However, DICER had no effect compared to the GFP control on the differentiation and hematopoietic development in vitro. So given that in the pure mouse, previously in the studies in the pure mouse models, they showed that the effects of DICER impairment on myeloid cells was only partial compared to that seen with DROSHA, and was had a more mild effect. We decided to then also have a closer look at the reconstitution of hematopoietic stem cell, differentiated human hematopoietic stem cells within our humanized mice that receive the DICER shRNA knockdown. And in these mice, we've showed that that DICER knockdown was having no effect on the frequency of DC populations within this mice compared to the reconstitution for the GFP control. It also had no effect on other myeloid compartments like the monocytes or lymphoid compartments with the B cells and T cells. Now, in the previous studies in the pure mice models that I was telling you about earlier, they showed that the severe effects that were shown by DROSHA deficiency on the regulation and differentiation of hematopoietic stem cells was mediated by specifically inhibiting the mRNAs. DROSHA's regulation of the hematopoietic cell difference was mediated directly by inhibiting specific mRNAs, specifically MIL9 and TODR1, by recognizing, and this was through recognizing the cleavage of stem loop structures within these transcripts. So we wanted to see if these targets were also the mediators of the DROSHA phenotype that we're seeing in our human hematopoietic stem cells. So firstly, we looked into MIL9 in our human hematopoietic stem cells. When looking at the two genes of MIL9, comparing mouse and human equivalents, we see a lot of homology in the 5' and open reading frames of MIL9 between the mice and humans. However, there is not a lot of homology in the 3' untranslated region between the two genes. So we had a look at, we went through and we had, in light of this, we still went through and we went through and looked at whether like was seen in the mouse studies, whether DROSHA deficiency had any effect on MIL9 expression in human hematopoietic cells. So we looked at quantitative real-time PCR of our human hematopoietic stem cells transduced with our different constructs. And what we saw in these studies is that with DROSHA deficiency, we got an accumulation of MIL9, suggesting that this is as was seen in mice, is a target of DROSHA, and that it's the deficiency in DROSHA is causing this particular mRNA transcript to be accumulated within these cells. So as part of Mark Schong's group, they then went on and looked at cleavage studies and mapping of cleavage sites within the human MIL9. And they, through these mapping studies, using a oligonucleotide adapter and PCR sequencing, they've identified 5 different sites that were able to be cleaved by DROSHA in human MIL9. And this is in contrast to in mice, where a single cleavage site was identified in the mouse equivalent of the MIL9 gene. They also showed through these studies, using these quantitative real-time PCR of using this adapter, that DROSHA deficiency caused a decrease in MIL9 cleavage products that were able to be detected by quantitative real-time PCR compared to our GFP control, confirming that DROSHA in fact is responsible for cleaving MIL9. So in light of these response, as was done in the mouse studies, we, in collaboration with Mark Schong, had used, they generated some overexpression constructs for MIL9 that we virally transduced into our human CD34s and engrafted into our humanized mouse model to see if the overexpression of MIL9 in this concept could replicate the impaired hematopoietic development phenotype that was seen with DROSHA knockdown. And so when we did this and analyzed our humanized mice and their immune reconstitution with MIL9 overexpression, we unfortunately saw that there was no significant effect on either the overall human CD45 reconstitution within these mice compared to controls, or in the specific subset development. So we saw no difference between GFP controls and MIL9 overexpression in the DC subsets or in the monocytes B cell or T cell development. And this suggests that suggesting that at least MIL9 alone is not able to recapitulate the effects seen with DROSHA deficiency despite it being a target of DROSHA cleavage. We also moved on to looking at TODR1. So TODR1 was, as I mentioned earlier, was identified in the mouse study as a second target of the DROSHA mRNA cleavage. Regulated DC development in a pure mouse model setting. Now, in mice, TODR1 gene is located on chromosome 16 between the genes of SDF22L1 and PPL2 genes. And this produces a open reading frame of around of 918 nucleotides. Now, comparative analysis for conserved sequences in the human genome revealed no equivalent conserved sequence to this to mouse TODR1. However, in humans, in the human genome on chromosome 22, there is a gene, an unidentified but annotated gene sequence that between SDF2L1 and PPIL2 on this chromosome. However, this produces a much more truncated open reading frame, only 331 nucleotides, and shows no resemblance to the TODR1 gene that was identified in mice. And when looking at DROSHA deficiency, looking at the DROSHA knockdown and comparing this to the GFP controls and DICER knockdowns, in human hematopoietic stem cells, we saw no upregulation of this transcript in the human hematopoietic stem cells following DROSHA knockdown. Therefore, the conclusions from this part of the study is there appears to be no human equivalent for TODR1, for the TODR1 gene. So the conclusions from this study showed that DROSHA is critical for the function of human CD34 hematopoietic stem cells, but not DICER. It also showed that as both DROSHA and DICER deficiency impaired the expression of microRNAs, that is unlikely that the microRNA pathway is essential for this function and for the development of dendritic cells in a human context. It is more likely in this case that the DROSHA's regulation on hematopoietic cell differentiation is being mediated by a microRNA independent function. We also showed that in humans, DROSHA is actually able to cleave MIL9. However, this cleavage of MIL9 alone does not appear to be the requirement for the cleavage of MIL9 and TODR1, of which we found no human equivalent mRNAs, does not to be the requirement for the cleavage of these mRNA targets is not required in humans for the proper hematopoietic development, and is not conserved between human and mice. The other conclusions that came from this study is that it showed that we are able to successfully use lentiviral constructs to manipulate the developing human immune system within our humanized mice by transducing the hematopoietic stem cells that we graft into these mice. And it shows that they are an effective model for studying mechanisms of human immune development. And it also really highlighted and helps really show that to the research community that the inconsistencies when trying to translate immune function from established mouse studies into a human setting. And we hope that these models will help pave the way to providing a model with a more relevant context for these studies. So I'd like to thank firstly my cancer immunotherapies group led by Professor Kristen Radford, and also Associate Professor Mark Chong's group in St. Vincent's Institute for all of their work with and support for this project, as well as the TRI flow cytometry core facility, BRF core facility here at TRI, and the Queensland Cord Blood Bank, as well as all of our funding bodies. And I would also like to thank all of you for listening today, and I would be happy to take questions.